I cannot be more excited today to introduce our speaker for the Pioneers in Biomedical Research seminar. The mic is on, yeah, okay, I'm gonna stay close to it. And so, yeah, some of you are wondering, okay, this is Neuroscience talk, so why is Sami introducing the speaker? So as of last week, I switched my field to neuroscience. I don't do cancer anymore. No, just kidding, no. All right, Cris and I met when we were both postdoc at UCSF, but let's step back a little bit, and I'm going to give you some background about his exceptional young career. So Cris Niell completed his PhD, and I'm reading a lot of things because there are just a lot of things on your CV, so yeah. But he completed his PhD in the lab of Steve Smith at Stanford. And there he developed innovative two-photon imaging of zebrafish optic tectum circuit developments. And there he was really successful as a PhD student, got an award, huge predoctoral fellowship, and published in Nature, Neuroscience, and Neuron. And so there from his PhD, he just moved a little bit north, San Francisco, not too far, and he did his postdoc in the lab of Mike Stryker at UCSF, and he became really a pioneer in the use of mice as a model to study visual processing and the development of neural circuits in the visual cortex. And so that's where we met. Chris's now wife, Denise Piscopo, was a postdoc in the lab where I did my postdoc, and so one of my first meetings with Cris was, Denise and I work in this beautiful new lab with a lot of windows, and then on the same floor, going through different turns, we were accessing a dark room where Cris was spending like many hours. And so entering this room, you had, of course, a lot of equipment. It was all dark, and then spotlight on this tiny mouse attached by the head with, of course, electrode in the brain. This is neuroscience. And then this mouse attached by the head, just running on the styrofoam ball suspended in air. And so I think that was not my first exposure to neuroscience, but I think that was a pretty good one. And so Cris was, of course, very successful in his work at UCSF. He got an NIH Ruth Christian NRSA postdoctoral fellowship and a Helen Hay Whitney Foundation postdoctoral fellowship. And he kept publishing in high-impact journals such as Nature and Neuroscience. So then it was the time for Cris and Denise to move away, and so Cris had like several offers and decided to go to start his lab in Eugene, University of Oregon. And so at that time, I remember thinking, why Eugene? And then 3 years later, I came to Roanoke, and my friends were thinking, why Roanoke? so I think we get pretty obnoxious when we live in San Francisco for so long. But what I want to say is that we talked yesterday, and Eugene and Roanoke are pretty similar cities, small airports. And I think this is great places to really, move forward in your career. You get a lot of support from the university and the community, and so that's not always the case in big university like UCSF, where it can be more competitive with more pressure. So since he started his own lab, Cris has built on this work to understand large-scale cortical processing from the level of individual neurons up to brain-wide pathways, maintaining a focus, of course, on visual processing. He was right away awarded with the prestigious NIH DHHS New Innovator Award just 1 year after starting his lab. Since then, he got additional NIH funding, R1, R21, as well as other supports. He kept being extremely prolific. In the past 8 years since he started his lab, he published 18 papers, including papers in Neuron, Current Biology, PNAS, Journal of Neuroscience, and Cell Reports. So Cris, thank you so much for visiting us. I hope you enjoy your stay. Sorry for the weather today.
Oregon.
Yeah, okay, good. At least you had like a good weather when you arrived yesterday. It's fantastic to have you here, and so looking forward to hearing about your research.
Thank you. Well, thanks, Sami, for the wonderful introduction, and thanks for the invitation to come out here and tell you about the work that we've been doing studying neural circuits for vision and action in the mouse. So when I got started studying vision, I thought of it as kind of this passive image processing problem. You look out at the world and you have to detect blue and green pixels to be able to identify objects, see where you are in the world. But vision is actually a much more active process than that. Sometimes you're not letting any sensory input come in. Other times you're solely processing one specific input coming in. So the brain has a shift how it's processing vision depending on your behavioral state. Likewise, you can do lots of different things with your visual system. You can track a fast-moving object in order to be able to drive a motor output, or you might be analyzing fine detail to make a visual discrimination. So you can use it to drive different types of behaviors. So my lab is interested in these three problems. How do you process an image, process the images coming into the visual system? How does that depend on your behavioral state at the time? And then how do you use that to be able to drive different types of behaviors? This is a quick overview of what I'm going to talk about today. I'll start off by talking about visual processing in the mouse. Can you study vision in the mouse and the effects of locomotion? Then I'll talk about some of our work looking at brain-wide circuits that are underlying visual behaviors, and then end with some more recent work looking at natural visual behaviors in the mouse. Okay, so visual processing in the mouse. So you might wonder, why are we studying vision in the mouse? Mice don't have great vision. The real answer is because of all of the genetic tools that are available to be able to study neural circuits. They kind of fall into two categories. One is the ability to fluorescently label cells. So we can look at individual neurons, see their synapses and wiring. This is actually an individual neuron in the visual cortex of an awake mouse, where we've labeled all the dendritic arbors in the synapses. So we can look at how we can observe the wiring and see how it changes with things like learning. And then also we can use fluorescent labeling to be able to measure the activity of neurons. This is an example of calcium imaging in the mouse using a fluorescent recorder of neural activity. Again, this is several hundred neurons in the mouse visual cortex, an awake mouse, and we can go in and actually watch patterns of activity across neurons. Then, of course, the other advantage of genetics is the ability to do functional manipulations, and in particular, over the past decade or so, the technique of optogenetics, which allows us to express a light-sensitive ion channel in neurons, shine light on those neurons, and activate them or inactivate them to be able to determine what their role is in visual processing. So this is why we might want to study vision in the mouse, but you might wonder, well, can you study vision in the mouse? Aren't they blind? Well, so then this was kind of the question that I started off with when I was a postdoc back at UCSF. What types of visual processing are conserved in the mouse? It turns out they do have very poor vision. It's very low resolution. An image like this ends up looking like this in the mouse. This is largely due to the fact that they just have small eyes and fewer photoreceptors. So it's essentially like having fewer megapixels in your digital camera. But the question at the time was, even though they have this much blurrier vision, are they processing the visual information in the same way? Are they doing the same computations, but maybe just based on lower resolution information coming in? So to get you oriented to the visual system, this is our visual system, obviously. Light comes in through the eye to the retina, goes up to the thalamus, and then gets sent up to the primary visual cortex. Most of what I'll be talking about today is measurements in the primary visual cortex. And mouse has a similar structural organization, retina, up to thalamus, up to visual cortex. So they have the same structures. The question is, are these structures functioning in the same way? So in higher mammals, particularly in cats and primates, studies going back about 50 years had revealed the fundamental properties of neurons in visual cortex. Work by Hube and Weasel going in and recording individual neurons found that neurons in the primary visual cortex don't respond just to pixels of light on the world, but they respond to edges of light at particular orientations. So this is an example. They present a bar at one orientation to get no response. You rotate it by about 30 degrees, the cell fires like crazy, and then it stops. So it's detecting bars or edges of light. And you can imagine that would be useful in processing a visual scene because it helps you, for example, extract the edges of objects that then allows you to go on and figure out what's out there in the world. So our question was, will the mouse visual system have the same property of orientation selectivity, detecting bars or edges at a particular angle, implying that it would be doing the same types of computations that a cat or monkey would be doing, the cat or monkey visual cortex is doing. So to approach this, I used electrophysiology. We back then started using multi-channel silicon probes. These are little electrodes that we can insert into the brain. This is the current version that has about 64 recording sites on it. Back in the old days, we were very excited just to have 16 recording sites. But the advantage of these electrodes is you can insert them into the cortex and record activity from a large number of neurons at once. This is an example showing 30 neurons. There's a couple advantages to this, one of which is just getting more data faster, which when you're a postdoc, it's great to collect data faster. But also now we can start to see the patterns of activity across neurons. And you can probably already start to see this. There's some of these neurons that are firing in phase with each other, and others that are firing out of phase. So we use this approach, put the electrodes in cortex, present visual stimuli, and see what the neurons are responding to. In particular, we used an approach that's called the spike-triggered average receptive field. This is a way to be able to map out what's the particular stimulus out there in the world, what visual image will cause neurons to fire. The way we do this is we show this random noise stimulus, just light and dark spots on the screen. Every time the cell fires a spike, we collect the image that was on the screen immediately before that spike, and we average these all together. And this will give you essentially what was the average thing that was on the average stimulus that was on the screen immediately before a cell fired. This is known as the spike-triggered average receptive field, and this is an example for one particular neuron. So we're showing all these white and dark spots, but on average, what was on the screen before a cell fired was a white bar with dark edges on either side. In other words, this neuron is responding much like you would see in a cat or monkey. It's responding to a white bar with dark edges at a particular angle. So we did this for a number of neurons. Here, this is color-coded now, so the dark spots will be blue and light would be white. But you can see the same thing here. We have a cell that's responding to an elongated spot at one angle. This is an edge between dark and light, and this is a dark bar on a white background. So these responses are much like what you might expect to see in cat or primate. In fact, we compared this to some data from Dario Ringach's lab that was recorded in monkey, and you can see that the responses are quite similar. The types of stimuli out there in the world that a neuron responds to were similar. One big difference is that this scale bar is 20 degrees, so mice are seeing spots that are about this big, and monkeys are seeing spots that are about this big. But you can see from the structure here, these look roughly similar. Now, you might think that I just chose the right examples and put them in the MATLAB color scheme, makes everything look the same. We were actually able to quantify this. We calculated a couple of scale-free parameters, and we were able to say that on average, the structure of the stimuli that neurons are responding to is similar despite this difference in the size. Now here I've plotted these two on the with different scale bars. If I actually plot them on the same scale, you can see how dramatically different the mouse and monkey visual system actually are. Mice are responding to very large stimuli. Monkeys are responding to a very small one, but they're actually quite similar. This is interesting for two reasons. First of all, it's saying that even though they're viewing the world at very different resolution, they're still extracting the same types of features out of the scene. They're doing the same type of computation, which means that there's probably something very common about how visual systems need to process visual scenes, even if you're operating with these different length scales. But for us, it was particularly exciting because it means, for the mouse and the monkey are processing visual information in the same way, we can now go start studying vision in the mouse using all of these genetic tools and have the hope that the underlying computations are going to be relatively similar. So our initial experiments were all done in anesthetized animals. And as I mentioned in the beginning, we wanted to see how visual processing depends on behavior in your state. And of course, there's not very much behavior going on in anesthetized animals. So we moved to the setup that Sammy described. This is a method developed by David Tank's lab at Princeton to put an animal on a styrofoam ball. Essentially, it allows us to be able to record from an awake animal that's held in place, yet have the animal be content. He can run. He can, in a minute here, he'll start grooming. But his head is held in place by this little metal bar. This is actually an electrode going down into the cortex. Over here, we would have a computer monitor to be able to present visual stimuli. The nice thing about this setup is we can record from the animal for several hours at a time. Doesn't even really realize he has an electrode going down into his head. But now we're recording from an awake instead of an anesthetized animal. So we can start to look at behavior. We realized, actually, once we started doing these recordings, at first we thought we were going to have to train the mice to do different types of things. But we found out relatively quickly that there was a big difference in the brain state just based on the animal's spontaneous behavior. You probably notice here he spontaneously alternates between sitting still and running. And when we started recording, we found that the visual responses were quite different in those two states. So this is an example of recordings from a single neuron. So time is along this axis. Each row is a repeated presentation of the same stimulus. Each of those little vertical bars is a spike of the neuron fired. And you can see that on each presentation, the cell fired a number of spikes. But if I color code them based on whether the animal is stationary in red versus moving in blue, this neuron fired about twice as many spikes when the animal was moving versus when it was stationary. This is true if we look at the peristimulus time histogram. This is the response as a function of time. You can see that there's roughly similar firing pattern, but that there's about twice as many spikes when the animal's moving. We can also see this if we look at the orientation tuning curve. We present a bar and see how it responds as you change the orientation. It's still responding to a bar at that same orientation, but with roughly twice as many spikes, twice the firing rate when the animal's moving. So it's really like they're kind of turning up the volume on their visual system when they start running. This was true across most of the cells that we recorded in the superficial layers of visual cortex. There was relatively little change in the spontaneous rate of these cells, but a big change in the visually evoked response. And this occurred on average with relatively little change in orientation selectivity. So it really seems like it's just turning up the gain on its visual system. So at the time, we were wondering, why would you turn up your vision when you start running? And our kind of cute just so story at the time was, well, an animal, when he's sitting still, especially if they don't have great vision, they may be listening to hear what's going on around them. And then once you start running, you need to be able to use vision to be able to navigate through your world, avoid running into things. And that was just kind of our cute story at the time. But it turned out when I moved up to University of Oregon, my next door neighbor, Mike Weir, who studies auditory cortex, did the corresponding experiment in auditory cortex. And in fact, auditory cortex does go in the opposite direction. That when an animal is sitting, actually, Mike chose the opposite color scheme that I did in mine. But when an animal is running, in red here now, the response is decreased. And when the animal is sitting still, the auditory cortex gets turned up. And the opposite thing happens in visual cortex. So it really is like the animal switching from auditory when they're sitting still to visual when they start moving. And since then, we found out it may be a little bit more complicated than that. For example, maybe that when an animal is using auditory, even when it's attending, you may want to be suppressing sounds because the animal tends to make, as an animal is moving, they make self-generated noise you may want to suppress. But it was clear at the time that the two, auditory and visual, are going in opposite directions. So there's a possible explanation of why the animal might be doing this. In my lab, we wanted to start looking at how this happens. Where in the brain does movement and vision get linked together? And we got intrigued by this area down in the brainstem, the mesencephalic locomotor region. This was actually inspired by a great MD Ph.D. student, Moses Lee. And this area, particularly the area around it, the pedunculopontine tegmental nucleus, is a target for deep brain stimulation. And when they, particularly in Parkinson's disease, and when they stimulate this area, it improves the patient's ability to move. But the patients also report feeling more alert. And Moses had seen this in the clinic and said, well, this is an area that seems to be linking together movement and alertness. Maybe this might be the one that sends a signal to the cortex telling it to turn up the gain. In fact, this area, I'll just call it the MLR for now. The MLR has both descending projections down to the reticulospinal formation and spinal cord that can drive movement, and then ascending projections to places like basal forebrain and thalamus that could then send a signal up to cortex to turn up the gain. So it has the right anatomical connections to mediate this. So we decided to take an optogenetic approach to turn on these neurons and see what the effect is on the animal's behavior and visual processing. So we used a virus to express channelrhodopsin down in the MLR. We lower a fiber optic down into the brain right above the MLR. And you'll see the effect over here. You might just barely be able to see the fiber optic, but when I turn on the blue light, you'll see the effect on the animal's behavior. So light comes on, the mouse starts running, light goes off, he sits still. Light comes on here again in a second. So this wasn't a total surprise. As I mentioned, the MLR has these descending projections that can directly drive motor output. In fact, if any of you remember the decerebrate cat experiments from your intro neurobiology course, this is the area that they stimulate to get a cat to start walking. So we kind of knew that this was going to be driving the motor output. The question is, does this area also have an effect on visual cortex? And you can see there's a little bit of a conundrum here because we stimulated locomotion, and I already showed that locomotion was going to cause changes up in visual cortex. But Moses had the insight that we could stimulate the MLR at a level that doesn't cause locomotion and then see what the effect is in cortex. So it turns out that if we decrease or we can dial in the intensity of the light so that at one frequency of stimulation we get no locomotion, and then we turn it up and we get overt locomotion. In fact, here the animal seems to tend to sit still, almost looks like they're paying attention if you want to anthropomorphize. But there's no overt locomotion here. So now we can study the effect of stimulating the MLR without the effects of locomotion. And sure enough, when we do this subthreshold stimulation, we see the same increased visual responses up in cortex. So this suggested a model where the MLR has these two outputs, one that goes down to the reticulospinal neurons and the spinal cord to drive the motion, and another pathway that can be activated at this lower threshold that could go and send the signals up to the cortex. In fact, Moses did further experiments stimulating specifically the projections to the basal forebrain, and these projections by themselves were enough to get the gain effects up in cortex. The basal forebrain is an area that expresses the neuromodulator acetylcholine, which is associated with attention, and other people, including Young Dan and Michael Stryker, went on to show that this circuit up to cortex then causes the increase in gain up in visual cortex. So we had a nice, neat little circuit that explains the locomotion. Of course, in reality, this circuit is much more complicated than that. I like this review from Laura Busey that shows at least part of how much more complicated it is. The MLR is also connected to the locus ceruleus, uses the neuromodulator norepinephrine, which is also involved in alertness. There's projections also of the thalamus, not just basal forebrain. So the circuit is much more complicated than our simple diagram. And I think in part that's because there's lots more things than just locomotion that can engage this circuit. For example, attention could act directly on basal forebrain to increase the gain here. And likewise, there's other things besides locomotion that can start to tap into this circuit, and then other things that can be activated. For example, locus ceruleus sends norepinephrine, which can dilate the pupil, involved in arousal. So even though we found one circuit, it's actually much more complicated than this. And there's another complication, which is, in our experiments, we don't know why the animal is running. We just put him on the ball, and he can spontaneously start to run or not. And the animal can run for any number of reasons. They can run to approach something. They can be running to avoid something. They can just spontaneously run. So we don't really know what the computation is that they're trying to achieve with their spontaneous running. So kind of a combination of how complicated this circuit is and not knowing exactly what it's being engaged for led us to want to search for circuits that are involved in very specific visual behaviors, and then say, how does this behavior get engaged, or which circuits in the brain does a specific circuit, the specific behavior engage? So then I'll transition into talking about our search for the brain-wide circuits that are involved in visual behaviors. So to do this, we wanted to have an animal that's doing a specific task. So it's not like locomotion where we don't know why he's running. Here we'll know what the animal is trying to achieve. So we set this up actually back at the end of my time at UCSF, working with Andy Huberman and Linda Wilbrecht to train a mouse in an orientation discrimination task, be able to tell horizontal versus vertical lines. So we know what the animal is trying to decide about its visual environment. The setup is shown here. It'll be a little bit more clear once I play the movie. This is an animal. He's in a clear acrylic box. There's 3 little nose poke ports here. What he has to do is poke his nose into the center port. These 2 stimuli will come up on the screen on either side, and he has to run over to the side that has the vertical bars, and he'll get a little water reward. So this is what it looks like in action. He pokes his nose, the vertical bars come up. Pokes the nose, vertical bars come up. Vertical comes up over here. So at the time, we were very excited. I've been studying visual cortex, showed that the receptive fields are similar to other species. But this was our first evidence that mice could do something complicated with their visual system. And in fact, they get quite good at it. They'll do several hundred trials in an afternoon. They get up to doing about 95% correct. And the nice thing about that, the fact that they do lots of trials and they do it very accurately, is we can now start to map out how accurate we can measure psychometric curves to determine how accurate their vision is. So we can do things like varying the relative orientation of those 2 gratings, making them closer and closer to each other, and seeing how their performance drops off. We can also vary the spatial frequency of the grating, how fine they are, to see the finest detail that they can detect, as well as measuring their contrast sensitivity. So in a way, this is a way to be able to give a vision test to the mouse to all see what can they see out in their environment. This is really nice, particularly for developmental studies, to be able to say, okay, if we mess with a developmental mechanism that we think affects, for example, orientation selectivity, and say how this affects their output in terms of ability to discriminate 2 orientations. However, what we've been using this more for is to be able to get the mouse just doing a specific task, not so concerned about how well they do it, but get them engaged in the task and see which parts of the brain are being used to achieve that, particularly in a case where we know, again, what the computation is that they're trying to achieve, such as orientation discrimination. So to do this, we kind of combine the 2 techniques of the head-fixed recording and this visual behavior. So now the mouse is on that same styrofoam ball, and he has to run on the ball towards the side that has the vertical grating. But of course, he's held in place. He runs to the left or runs to the right on his little running ball, and he gets a little water reward here from the spout when he does it correctly. But the advantage of being head-fixed is that now we can look into the brain and see which areas are being engaged by this task. So to do this, we use calcium imaging. Calcium imaging is a technique takes advantage of when a cell fires an action potential, calcium ions are going to rush into the cell. So if you have a fluorescent calcium indicator, it's going to light up when a cell is active. We use this one, GCAMP6, that was developed by Lauren Luger at Genelia Farms. And we made a transgenic mouse that expresses this sensor, GCAMP6, in all of the excitatory neurons of cortex. So if we look down on the top view of the brain, we can see the whole brain is glowing. But now when an area of the brain is active, or a particular neuron is active, it's going to light up. And rather than zooming into the single cell level, we started off by kind of zooming out to get a brain-wide view we built this custom macroscope that lets us see about 2/3 of the dorsal surface of the cortex at once, look down and see the pattern of activity. So this is what that imaging looks like. Up here in the corner, you can see the stimulus we're presenting. It's just this kind of flashing on and off white noise. But every time the stimulus comes on, you'll see this area here light up. This is the primary visual cortex. You might see a couple other little spots around there also lighting up. Those are the extrastriate visual areas. Now, as a visual physiologist, I would just tend to go stick my electrode straight into V1 and record what's going on there. But what was really striking for us, taking this bird's eye view, is how much other activity there is going on in the brain. This is a mouse that's just looking at this pattern on the screen. He's sitting on his ball. But there's a whole bunch of other areas that are being activated. Some of these are kind of obvious. This one is the barrel cortex. So every time the mouse starts whisking, that lights up. Up here is motor and then somatosensory, so when the animal's moving. But then others aren't so obvious. This is retrosplenial cortex, which is, actually I didn't know much about retrosplenial cortex until we started seeing it light up. But it's an area that's involved in kind of your sense of space in the world. And so there's all these areas that are active when an animal's just viewing a visual stimulus. And so we got interested in how does the visual areas affect what's going on in the rest of the brain, and likewise, how does everything else in the brain influence the visual processing. Now the nice thing, because we're looking at large areas of the brain, we can use this technique to map out what the different modalities are across the cortex. So we can take that movie and we can average every time we're presenting the visual stimulus, and we see the visual areas light up. We can average every time the animal starts whisking, and you can see the whisker areas light up, the barrel cortex. We can average every time the animal starts running. We see motor and the limb somatosensory light up. And together we can use this to parcel out the areas of the visual cortex and see which are involved in different aspects of what they're doing in their spontaneous behavior. Of course, as vision people, we do tend to zoom in on the visual areas. This is zoomed in on the posterior region of the cortex, showing this white noise. Now it's a bar moving across the screen. And as the bar moves across the screen, you can see the corresponding bar of activity moving across the cortex. And this was kind of like my dream when I started in neuroscience, to be able to look down the brain and literally see what the animal's seeing out in the world being mapped onto their cortex. So this large bar here is the primary visual cortex. Again, each of these other little spots that are moving out here are the extrastriate areas. We can map these out in the mouse. There's about 7 to 10 of these, depending on how you define them. They've been mapped roughly into a dorsal and ventral stream that corresponds, oops, sorry, I don't have that here anymore. In humans, there's also an idea of dorsal and ventral streams that are involved in what versus where processing. And one of the things we've been interested in is seeing how these different extrastriate areas get engaged during different types of behaviors. So in fact, we can put this all together now. We can combine our behavior with the wide field calcium imaging. In a sense, this wide field calcium imaging is like fMRI. We're getting this kind of global view. But the advantage is we have much better temporal resolution. We can get down to about 100 millisecond temporal resolution, and that allows us to see the spread of activity across the cortex as the animal's making a visual discrimination. So you can see here, there's a couple zooms in here. The activity starts in V1 in a couple of the extrastriate areas, spreads across the visual areas, and then you get this activation out here. This is actually the hind limb somatosensory area, and that makes sense because when the animal's making his response, he's going to move on the ball, and so he gets somatosensory input. So we can see the spread of activity from when he sees the stimulus to when when the animal's making the decision. So much like fMRI, we can use this to map out areas that are involved in a task and how they change over time. One of the things that we've done with this is to look at how the pattern of activation changes as an animal learns a task. So this is an animal that's naive. They've learned how to use the ball, and they know that they get a lot of rewards for making the right choice. But he hasn't been presented, or this is the first day that he's been presented with a specific task. For example, vertical versus horizontal. And then this is after about a month or two of training when they've learned to make that discrimination. There's many differences here. There's a couple that I want to point out to you. I should also note this is that sequence of activation over time. So about 500 milliseconds from when they see the stimulus to when they've made their response. One of the biggest differences is in the visual areas. And in fact, you see a decrease of the activation in the visual areas as the animals learn the task. This wasn't quite what we expected. We kind of thought, as they learn to do a visual task, they'll be using visual cortex more. But my colleague, Mike Posner, said that, of course, this has been known for a long time in the human literature, that particularly in cases of implicit learning, the activation in visual cortex goes down over the course of learning. But of course, with fMRI, you don't know the neural basis. What's changing that causes that activation to decrease? Another difference we saw was in this somatosensory. This is the primary and secondary hind limb somatosensory areas. And I had noted before that these are activated when the animal is making his response, but that activation is much stronger after the animal's been trained. So we think it's not purely the sensory input with making the response, but something about the trained pattern of sensory input that particularly drives these somatosensory areas.
Quick question. Could you, either in this or the previous slide, sort of roughly map out the monocular versus the binocular part? My question really is the training effect equivalently strong in both those areas?
That's a great question. So the binocular areas right here, the reversal between V1 and these lateral extrastriate areas, our stimulus is presented just lateral of the binocular zone. And so, or probably overlapping with the binocular zone and into the monocular zone. We haven't looked at that yet. There are interesting things, though, in the map, a little bit of divergent, but Bill Hader has shown that mice actually learn even a simple discrimination like horizontal, vertical, doesn't require any kind of depth or anything like that. They actually learn it better when it's presented in the binocular zone versus the monocular zone. So, but we didn't do anything here to test that. So we see this change in the somatosensory cortices. And the last one I want to point out is actually up here. This is auditory cortex. And auditory cortex is strongly activated when the mouse starts learning and then goes down. You might wonder, why is auditory cortex being activated in a visual task? Well, when we give the animal its water reward, there's a little click when the valve opens to give them their water. And so that click is called the activating auditory cortex. It's a reward signal. And then over time, as the animal has learned that, it becomes less salient, and the auditory cortex is no longer responding. So as I mentioned, this wide field imaging technique is much like fMRI. We can find where changes are happening. But the advantage of the mouse is we can now go in and we can say, what are the neurons doing? So we can choose an area that's changing, for example, primary visual cortex, and then go in and do calcium imaging of hundreds of neurons over the before and after learning to compare how these responses are changing. I'm not going to go too much into these results, partly because they're work in progress, but it looks like most of that decrease of the activation in V1 is due to a population of cells that respond transiently when the stimulus comes on and aren't encoding the visual information quite as properly. So we think there's an initial activation, something to do with salience or learning, is driving that initial strong response that then decreases over time. The other reason I don't want to go into too much detail about this is because as we were doing these experiments, we started to realize that this task isn't really what the animal's visual system is wired up to do. A mouse out in the wild never has to learn, vertical bars means run over to the right to get a water reward. It takes the animal several weeks to be able to learn this task. They sit there every single day saying vertical, horizontal, vertical, horizontal. All they have to do is run to the right and they'll get their water. And it still takes them 2 or 3 weeks to be able to make that association. So I think it's something that it's great to be able to study learning. How do you form those associations? But it's not that useful if you want to say, how does a mouse normally use its visual system to figure out what's out there in the world? So that led us to search. Oh, sorry, that's our task up there. That led us to our search for ethological visual behaviors. What does a mouse actually use its vision for? And can we figure out the circuits that underlie that? So what does a mouse actually use its visual system for? Well, one thing that's well known is fear responses. Looming stimulus overhead, for example, a predator, or something resembling a predator, will cause an animal to either freeze or to flee. And there's a number of groups who have been studying that, particularly the role of the superior colliculus. Mice also use their vision for navigation to be able to find their way around out here in the natural world. But I also like to point out that one of those standard tests that people use in mouse, say learning memory, the Morris water maze, the mouse has to swim over to a little platform to escape the maze. That's actually a vision task because the way they find their way to that maze is based on visual cues that are around the room. And for example, place cells are dependent on that visual input. So we know that they use their vision to be able to drive navigation. But my postdoc, Jen, got interested in whether they would do prey capture. And that was inspired by this species here. This is the grasshopper mouse. It's a species that's been described as being specialized for a carnivorous lifestyle. It has large claws and large teeth, and it can catch grasshoppers and even scorpions and frogs. And people always said they were specialized to be carnivores. And so we didn't necessarily think our lab mice would be able to do prey capture. And in fact, I didn't think so. Jen came to me one day and said, I want to see if mice will catch crickets. I said, oh, no, our mice are lazy. They just eat the food pellets that we give them. And they've been inbred for so long, they've never experienced prey. They aren't going to catch anything. But she did the experiment. She just stuck crickets in the cage. The mice go, and they catch it and eat it. And so then she was off and running. So she worked really hard on being able to quantify this behavior. She set up a little arena. It turns out it takes a little bit of habituation to get them used to a bright, well-lit arena with an experimenter standing over it. It's kind of stressful for a mouse. But once you get them habituated to the arena and make sure that they're hungry, in fact, they will hunt crickets. So this is the mouse here. This is the cricket over here. You see that once he sees the cricket, he makes that straight beeline straight to it. So what was exciting to us was these really long distance approaches when he goes all the way across the arena towards that cricket. Turns out when he gets to the cricket, he's not quite as good at catching it. And that may, that may be where that specialization comes in. They don't have the big teeth and big claws to be able to catch it. It's actually kind of good for us because it means we essentially get lots of repeated trials of the cricket gets away, and then he chases after it. And it turns out that that's the aspect that they need vision for. So Jen set out to test which aspects of this require vision. She did this first by deafening the animals, by putting little earplugs into their ears. And you can see that when we do that, the mouse is just as good at doing these long-range approaches towards the cricket. So they aren't impaired by having their hearing blocked. There's a couple of controls that we did for that as well that I can discuss. However, she put them in the dark, in fact, putting them under near-infrared illumination so that we could record them. You can see that the mouse doesn't really seem to realize that the cricket is there. Wanders right past it here. But fast forward through a minute or so of them wandering around the arena together. You'll see that then, once the mouse bumps into the cricket, so essentially once it knows that the cricket's there, it knows roughly where it is, then they can chase the cricket and catch it. And it turns out that they need hearing to be able to do this phase of it. If we put them both in the dark and earplugged, then they won't catch it at all. But it seems that they need vision for these long-range approaches, and then they can use hearing once they're up close. There's a couple explanations for that. One may be just that the cricket's making a lot more noise when it's being chased. It may also be something about them being better at localizing the cricket using auditory cues when they're much closer to it.
So it almost, it seems, it's not quantified there, but it seems like once he knows the cricket's there, it's just about as good as he was in the light with just the auditory system. So as if the visual cue is the initial transient trigger, there's a cricket.
Yep.
And then he can just go heavily to.
Yeah, that's exactly, and I'll actually come back to that in a minute when we get to the neural circuits. But yeah, we think that the vision is for telling that it's there and then doing this long-range approach across the distance. And then once he's there and in contact with the cricket, then he can use other things to be able to catch it. So Jen quantified this. This is looking at the angle of the mouse's head relative to where the cricket is, the azimuth. And you can see when the animal's in the light, they aim quite accurately, about plus or minus 10 degrees. It's relatively similar if we earplug them. If we put them in the dark, then they fall almost to chance. This residual bump here is from exactly those cases when they bumped into it. Then they can still orient towards it. And then if they're earplugged and in the dark, they essentially have no idea that the cricket's there. I really like this graph that Jen made, which is looking at that accuracy, how accurately they're targeted relative to how far the cricket is from the mouse. Because it tells us a few things that we'd always wondered about how the mouse uses its vision. One of which is looking at, well, first of all, you can see when they're in the dark, they never start orienting towards the cricket. And when they're in the light, starting at about 15 or 20 centimeters away from the cricket, they start orienting towards it. And this answered one of the questions we had is, what's the operating distance of the mouse's vision? They have these really big receptive fields. You could use that to see something that's really big but far away, or maybe use it to see small things that are close up. And this told us that mice can use it also in this kind of intermediate range of 15 to 20 centimeters. You can also, the other thing is that when the cricket is about 15 or 20 centimeters away, the cricket's about 1 centimeter across. And so that tells us how large an object that they can detect. At 15 centimeters, the cricket is about 5 degrees across. And so that suggests that, and that's roughly the size of a retinal ganglion cell receptive field. And so that suggests that mice are actually able to detect things that are about the size of an individual receptive field. And then finally, when they're actively pursuing the cricket, they're orienting within about plus or minus 10 degrees. So this is even as the cricket's moving around trying to get away from it, they're able to aim to within plus or minus 10 degrees, which tells us that they're able to convert that visual input into a motor output that's almost as accurate, again, as the retinal ganglion cell receptive fields. So this told us, essentially another way to be able to give the mouse a vision test, but in this case, it's relying on their natural behavior instead of these trained behaviors. And the nice thing is the mice kind of do this innately. You can put them in there, and they'll do it without several weeks of training. And we also hope that this will be tapping in to the natural circuits for vision. In fact, that led us to start look at what are the circuits in the brain that are mediating the prey capture. And for this, we moved to look at another brain area, the superior colliculus, This is an area down in the brainstem. You guys might have heard about it from Mike Fox's work. It receives direct input from the retina. And in us, it's thought to be mostly involved in, it's involved in reflexive action. So, for example, if something moves into your visual field, you either turn towards it or you move away to duck away from it. And in fact, the superior colliculus is really ideally suited for that, converting a visual stimulus into an action, because it has overlaid maps of the sensory world and the motor output. So one location on the superior colliculus will respond to a visual input at one place, and right below it are motor neurons that will cause a movement to that location in space. However, superior colliculus is also interesting because recent evidence is suggesting that it's involved in humans, not only in these kind of overt movements, but also in directing your attention to a particular region of space. So we're kind of hoping that by studying the superior colliculus in the context of prey capture in mice, might tell us something about how these circuits have then been co-opted in us to be able to mediate orienting of attention. So to study this, we've been taking advantage of a couple of Cris lines that were discovered by Gabe Murphy. These access two cell types that had actually been defined about 100 years ago by Ramon y Cajal, the wide field vertical cells and narrow field vertical cells. And these two cells seem almost ideally suited to two different aspects of the prey capture, the detection of the prey and then the approach towards the prey. In particular, the wide field cells have these really big dendritic arbors. They have correspondingly large receptive fields, and they detect a small object anywhere within that receptive field. So they're good at detecting things, but not necessarily localizing them within the visual field. And then they project to LP, the mouse pulvinar and thalamus, which could then send a signal out to the rest of the brain, detecting that something's there, and then switching into hunting mode. On the other hand, these narrow field cells have very small dendritic arbors, correspondingly large, small dendritic arbors, small receptive fields. They're also sensitive to the direction of motion. So you can imagine they'd be ideally situated to be able to say there's an object there that's moving in this direction. And then they project down to the deep superior colliculus, which is where those motor output neurons are. So they can convert that, there's an object here out in space, and convert that towards a motion towards that location. So to test the role of these cells, detection versus orienting, we use a chemogenetic approach. This is an approach where we can use a drug to suppress the activity of each of these cell types individually based on Cris expression. We do this using a virus injection of these DREAD molecules to express DREAD molecules. For those of you who are familiar with DREADs, there's some caveats about CNO that I'll be happy to discuss at the end if you want. But what this allows us to do is to selectively suppress these cells over the course of a couple of hours and measure the animal's prey capture performance. So an example of shutting down the wide field cells is shown here. Mouse is right here, cricket's over here. Normally by this point, the mouse would have realized the cricket was there and started approaching towards it. Here, eventually he realizes it's there. He still makes that direct approach. But then when it gets away, it's almost like he doesn't realize that it's still there. Then he notices it's there, he makes that approach. So this looks like he has a deficit in the ability to detect the prey. And in fact, we found that these two different cell types did have distinct effects on the animal's performance. This is showing the frequency of making approaches compared to the wild type. Both the wide field and narrow field cells have deficits in starting these approaches. With the wide field cells, it's more a deficit of noticing that it's there, whereas with the narrow field, particularly when the cricket is at a distance, with the narrow field cells, it's that they don't turn towards the cricket. And then once the animal has started making his approach, this shows his probability of intercepting it, successfully intercepting the cricket. And you can see that for the wide field cells, once they've realized it's there, they're still pretty good at getting to it, whereas the narrow field cells have major deficits in actually being able to aim towards the cricket and actually reach it. So we think these cells are involved in the detection. These are involved in the orienting. As I mentioned at the beginning, when I first started talking about superior colliculus, there's ideas that it might be involved in attention. And in fact, again, my colleague at Oregon, Mike Posner, classically described attention as having multiple phases: alerting, orientation, alerting, and then orienting. And so we think that this, alerting, realizing something's there, and then orienting, turning your attention towards it. And so we think that perhaps these are the cells that may have been co-opted to mediate those different aspects of attention. So we've been using this chemogenetic approach to shut down the cells to be able to see what their role is in behavior. But we'd also like to be able to record from these cells and say, well, what are they actually doing during the prey capture? But there's a problem with that, particularly for a visual physiologist. What we want to do is we want to say there's a visual stimulus here in space, and what's the response of a neuron that we're recording from? But of course, as the animal is doing prey capture, he's running around the environment. His head is moving all over the place, so we don't know what he's looking at. But also, his eyes are moving at the same time. So we need to solve this problem of where are they looking, where is their head pointed, but also where are their eyes looking within that. So we've been taking an approach to try and measure this using a set of head-mounted cameras that we place up on the mouse's head. This takes advantage of the fact that miniature cameras have become very cheap and accessible recently. It's essentially the same cameras that are in your iPhone. So for $25, you can buy a little mini camera that we can then mount up on the animal's head. We have one that looks out at the world, so we can see where they're aimed. Another one that's looking back at the eye. Either in some of our setups, we have it looking directly back. Sometimes we have a little IR mirror to reflect the image back. And so then using these two, we can figure out, first of all, where are they looking in the world, and then where is their eye pointing by measuring pupil. And eventually our goal is to be able to add a silicon probe into this, although we aren't quite there yet. But we have the other parts working of being able to look and see where's the animal's eye pointing and where they're looking at. This is an example here as an animal's exploring an arena. You can see its eye movements here. So you can see how frequently and how large the eye movements are. So clearly we have to compensate for those. And what the visual input is in terms of the scene that's in front of them. So we're going to just get going with this, but we're really excited about the idea, first of all, to be able to figure out how the animals are moving their eyes and their head as they go through the world, and then being able to link this up with neural activity, and essentially being able to release this bond that we've had of always having to hold the animal's head in place to be able to do visual physiology. We want to study these types of natural behaviors to now be able to allow them to move through the world naturally. And I actually hope this will kind of answer one of those questions I brought up way back at the beginning about locomotion. That we don't always know, when an animal's stuck there on a ball, we don't know what they're trying to do when they start moving and running on the ball. But now if we can have them undergoing locomotion for a reason, whether it's exploring or catching a cricket, we might be able to make a little bit more sense of some of those circuits involved in locomotion. So if I have a couple minutes left, I might switch to tell you something about completely new that we're doing in the lab, which is studying the octopus visual system. I made an argument at the beginning, we're studying the mouse because it's similar to the primate and presumably our visual system. We're actually studying the octopus because it's so completely different from our visual system. We diverged from cephalopods and vertebrates diverged about 500 million years ago. Our common ancestor probably had photoreceptive cells, but nothing resembling an eye. Yet we both evolved these very elaborate and quite remarkably similar camera-like eyes. So octopus has very good vision, high acuity because of this camera-like eye, but it's connected up to a brain that's just totally weird. It looks like a giant slug brain. In fact, even in the scientific literature, it's often referred to as an alien-like brain. But it's actually great for studying vision. An octopus's brain, adult octopus brain, is roughly the same size as a mouse's brain, but about 2/3 of its brain is these optic lobes that are devoted to visual processing. And they perform a large number of really interesting visual behaviors. Everything, they can do prey capture also, they can do observational learning, they can use visual input to match the camouflage on their back. So we're interested in is how do they use this remarkable sensor to then be able to process, and how does the information from this sensor get processed by this totally different type of brain? And what's kind of remarkable to me is that very little is known about this. So in contrast to the primate or mammalian brain, where we kind of know how visual signals are transformed, what are the brain areas and how are the signals being transformed, or in the fly visual system, we know how a lot about how color and motion are processed. In the octopus and cephalopods in general, there's very little known about the neural coding. There's been recordings in the photoreceptors, but there's been no recordings of visual response properties in the central visual system of the octopus. So we have no idea whether, for example, they'll have things like simple cells like we do, will they have direction selectivity, orientation selectivity. And of course, even less is known about how this is used to drive different behaviors. There's been a lot of anatomy that's done, so we know from lesion studies which parts of the brain are involved in things like learning and memory and camouflage. But we don't know the information being processed at each stage and then being sent out to the brain areas for these different types of behaviors. So to get into this, we've started studying juvenile octopuses. It makes the experiments a lot easier to work on small animals. Let's see if this is going to play. Oh. Okay, here we go. So this is a juvenile. His body is about the size of a pea. The arms are a couple of centimeters across. This is the postdoc Judith dropping some brine shrimp in there. You can see that they do prey capture. You might have also noticed that when the brine shrimp first came in there, it suddenly changed his camouflage pattern. He went from being dark to being light. These movies are kind of captivating to watch. Different arms are doing different things. Yeah, one arm's going back into his home, and another one's reaching out here to grab brine shrimp. So eventually there'll be some remarkable behavior to be done. Although as we were talking earlier, there's still challenges to free-moving octopuses. But due to the difficulties in recording from the octopus visual system, and I should say, it wasn't for lack of trying that people haven't recorded from the central visual system, but it's actually just quite hard to record with standard electrode techniques. So we decided to take a calcium imaging approach. This, it turns out, is actually very similar to work that I had done back during my Ph.D. Studying the visual system of zebrafish. And what we can do is we can take the animal. In the octopus, there's no genetics. We can't do GCamp yet, so we have to inject a calcium indicator dye. We inject the dye, then we can we do some dissection to expose the optic lobes, affix them to the bottom of an imaging chamber, and then we can present visual stimuli on the side of that imaging chamber and measure the visual responses. So this is an example of one of our first calcium imaging experiments. In a second here, we put, fortunately contrast is a little bit low. Oh, there you go. In a second, you can see sets of cells lighting up when we present the visual stimuli. But much like we do in the mouse, we can record from large numbers of neurons at a time. We can measure the neural activity and then relate that to visual stimuli that we're presenting. So this is still relatively early, but we have shown one finding about the octopus visual system, which is that they do have a retinotopic map. So much like our visual system, where there's a map of the visual field that's represented at multiple stages, including a map out on visual cortex, there's a smooth representation of visual space out on the octopus optic lobe. So this is showing stimulus presented at the bottom of the screen versus the top of the screen. Each of these dots is an individual neuron that we recorded, showing its preferred location within the visual scene. And you can see that there's a smooth progression along the vertical axis and along the horizontal axis. So they do have a retinotopic map. Maybe not completely surprising because the axons coming in did seem to be retinotopically arranged, although there are other species like turtle cortex that don't have a retinotopic organization. So it's kind of nice to be able to confirm this in the octopus. We've also started to look at some other organization response to other response properties. This is what I showed you a second ago. This is retinotopy. So if we present a stimulus in the center, these set of cells light up. You move the square over a little bit, a separate set of cells light up. It turns out if you present a spot in the same location but switch from being dark to light, you also activate a different set of cells. So the response to the dark spots are here, and the response to the light spots are here, suggesting a segregation of on and off processing. So as I mentioned, this is still a relatively early project, but our eventual goal is to do the same types of things that we do in the mouse visual system, or people have been doing for 50 years in the primate visual system, to be able to map out the responses to different types of stimuli, whether simple stimuli that we use in kind of standard visual physiology stimuli like gratings and moving spots, white noise. We're starting to look at natural scenes and see if there's differences in the statistics of the underwater environment. A neat thing about Octopus is they can detect the polarization angle of light, so we'd like to see how that's encoded in the brain. But then eventually be able to link this up into the circuits that are driving different behaviors. So you'll be able to say what information is extracted by different cells, and then get what gets sent out to different areas that are involved in different types of behaviors. So with that, I'll finish up and thank all the people who have been involved in this, all of the folks in my lab, my collaborators, and funding sources. This is Eugene, Oregon, which actually, to me, reminds me a lot of Roanoke when I flew in yesterday. So maybe get a chance to come out and visit us one of these days. Thanks a lot. Yeah.
So all your tasks in the mouse were largely involving objects, patterns, and maybe prey. They're social critters. Have you ever explored whether or not vision plays a role in social interactions as cues?
We haven't done that yet, although that's actually, so the free-moving animals with the cameras, we're really interested, there's kind of these unsolved questions of how does an animal without a fovea want to move its eyes and head in the world? Like if you don't have a fovea, you don't necessarily have to move your eyes to get higher acuity vision. It seems like a lot of their eye movements are compensating for how their head moves. But we think there'll be interesting things about, that you can't always do compensatory eye movements. You would never look at anything new. So we can start to see when they're looking at a new object by when they kind of break that compensation. I think that's going to be the equivalent of a head saccade. So again, we don't know when they're going to do those types of movements. And so we've been talking about, like, okay, what kind of environments can we put them in where they might be interacting with different types of objects, and how are they seeking out information in those different contexts? I showed the novel object recognition, but one of the ones we've been talking about is social behavior. If you put a mouse in there with a conspecific, or maybe same or different sex, how do they interact with it, in terms of, are they looking at it, are they looking away from it? But we haven't done that yet. Yep.
Yeah, for the prey capture stuff, do you think that would this even be possible to train the mouse to associate an auditory cue with placing the cricket in the arena to mimic that kind of initial orientation towards it and then put them in the dark? Do you think that they would still have a defect in orienting towards the...
Yeah, so that's actually quite interesting. So we started off this project with my next door neighbor, Mike Weir, who studies auditory cortex. And at that time, we weren't sure whether they're going to be using vision or auditory. Our setup is a little bit skewed towards being vision. It's a bright, well-lit arena. The cricket actually doesn't make that much sound. And so vision ends up being, especially for those long-distance approaches, if the cricket's sitting still, there's no sound to know where it is. Mike's actually shown that if you raise an animal in the dark, and they only experience crickets in the dark, and he drops the cricket in, so it makes a noise when it lands, and they know where it is, in that case, they will use auditory for it. And Mike was actually telling me this morning that he has, mice that basically don't have any vision can still do it. Probably, if you've grown up blind, you develop other strategies to be able to do it. Yep.
Okay, so quick mouse question, a quick octopus question.
Okay.
So on the mouse, as I was watching the movies in the dark, and there were earplugs, and I watched them converging, I got a sense that the trajectory was sort of relatively even velocity. That is, they didn't seem to be compensating when they got close. So my question is about disparity detection, engaging depth. And so you just actually were alluding to the fact that, as I understand it, they have somewhat relatively disjunctive independent eye movements. And so when you start to look for disparity detection in the visual cortex, that's kind of a rough thing to do. So that's my question. Are they using that? And are they using that as a depth cue?
But yeah, and so actually, my grad student had just acquired a whole bunch of data with the eye cameras. In fact, we switched from having one out of the world to having two aimed back at the eyes during prey capture, so we can kind of address that question directly. So yeah, they have very disjunctive eye movements, and it kind of makes sense. If your eyes are on the side of your head and you're trying to compensate for head movements, if you tilt your head this way, your eyes have to move opposite directions. If you move your head this way, they move in the same direction. Put that together, you get eyes that are going all over the place. And so how you get binocular fusion out of that is a big question. There's been ideas that maybe once you're hunting something, you actually kind of lock into it. And in fact, in zebrafish, they've shown that, and actually cuttlefish also. That the eyes kind of, when they start to do prey capture, in fact, the cuttlefish basically creates a binocular zone right before it strikes by converging its eyes. So it may be that in those cases, they lock their eyes onto a target, but we don't have the answer to that yet.
Well, thanks.
In fact, my grad student just sent me a movie last night that I almost put in here of the two eyes during prey capture. But.
So my quick, if it could be octopus question is, you obviously are sitting in a very powerful position. Maybe you already know the answer, but I'm curious. With the 8 arms out there capturing the brine strip independently to address the question of true parallel processing. So my question is, are they sequentially making a computation for where brine chip X is for arm number 2 and their brine chip Y for arm number 5? Or are they truly those circuits operating in parallel in real time to solve both?
Yeah, so I don't have an answer to that, but the octopus definitely is viewed as kind of a model for distributed processing. Actually, about half of their neurons are out in the limbs, and there's a lot of complex circuitry out there. So it's thought that there probably is, there's probably a master plan that's coming down from the central brain, but then the arms act independently to do a lot of that. In fact, a severed arm can do very complex motor behaviors. There's a little bit of, they have a neat thing, if you have a floppy arm like that, it's actually relatively hard to control. It's a hard motor control problem. And it turns out they actually create joints in their arm using hydrostatic pressure that kind of reduce the degrees of freedom for that. And they'll do a lot of those things, in the absence of a central brain. So we aren't studying that, but definitely for soft robotics, there's a lot of people who are interested in that motor control aspect of it as well. Yeah.
So I thought what you showed for the wide field cells versus the narrow field cells is really cool. Have you thought about doing the same for the ganglion cells? What ganglion cells are required for this function? Because at 20 centimeters, I would imagine that they're really detecting movement more than the acuity of a cricket. And you could probably test that by putting in a cold cricket that's not going to move or something.
Yeah, so it turns out they will go for a dead cricket. I don't know if we measured quite how accurately or the latency to approach to it. They will still catch it. But you can also imagine as you're moving your head, you're creating motion on the retina. So we haven't done that per se. I know Carl Farrow is characterizing the retinal ganglion cells that provide input to each of these different cell types, and so that'll probably provide some of the answers. But yeah, we haven't started doing that yet. Yeah.
So you showed that locomotion changes sensory processing. In auditory system, you will have attenuated response, and in visual system, you have enhanced response. Is there any idea about mechanisms, whether those are local circuit mechanisms or like global neuromodulator secretion?
Yeah, so actually David Schneider in Rich Mooney's lab has done some nice studies of auditory cortex. And there a lot of it does seem to be a direct projection from motor cortex into auditory cortex that's providing feedback and suppressive. And in fact, Dave Schneider has done nice things showing that probably a lot of that is suppressing self-generated noise. So it actually has a mouse that, every time it steps, they make a little clicker. They have a speaker that makes a click, and then over time the response to that, even though the mouse hasn't generated, because it's paired with their movement, they learn to suppress it.
So it's kind of lateral inhibition sort of phenomena, or?
Or a predictive coding, subtracting a predictive signal based on your motor commands. It is interesting because I think in the vision side, people have been looking at it more from the neuromodulatory perspective in acetylcholine, and the auditory side, they came at it with this idea of predictive coding and the motor control. And nobody's really quite done the opposite experiment. My guess is there's some of both. In fact, my guess is that a lot of what these locomotor signals are is when you move your head through the world, you need to compensate for that in your visual system. And that when an animal's head fixed, they're trying to move, and maybe you're trying to create those signals, but then the visual world isn't moving the way it should to counteract those signals. We know it's not a pure mismatch signal because we did the test of, when a mouse starts running, we played gratings of different orientations. So if it was a mismatch signal, then if gratings going this way would match your movement, gratings going this way would mismatch, and we don't see a bigger signal for that. Although actually, other people have done it with like closed loop virtual reality, and if you break a virtual reality, then you do start to get mismatch signals. So that's part of the reason why I said, we found one circuit that mediates this, but I think there's a lot of other things that tap into it, particularly in different contexts. Yep.
So, Asthma, so when you stimulate, you find the multiple phase in the neural activity. Do you ever see a specific neural activity also change?
Oh, with locomotion?
Yeah.
We haven't, but I was talking to somebody, Matteo Chiarini's lab has some data on that. I don't think anybody's yet published motor effects and locomotion effects in superior colliculus, but they're definitely there. A couple groups I've heard about recently, but nothing published yet. And I should also mention there are some effects in LGN. They're much weaker than they are in cortex. And part of this, I think, is just because the brain is interconnected. So if V1's doubling, that's going to trickle out to other areas as well. But there's also probably some direct effects on those.
All right. Please join me. Thanks.
Thanks.
Cris.